you. Good morning, everybody, and welcome to the first edition this year, the January training. Today, our guest speaker is Joe DeMark, PhD, Corteva Agriscience. He's a field scientist from Fayetteville, Arkansas. He's originally from Racine, Wisconsin. He has been a research scientist with Dow Co., Dow Agrosciences, and Corteva Agriscience for 30 plus years. In '85, he graduated magna cum laude from the University of Wisconsin-Whitewater with a BS degree in biology and a minor in chemistry. After earning his master's and PhD degrees in entomology from Purdue in 1988-92, respectively, he joined Dow in 1992. Throughout his career, Joe has worked in urban pest management, research and development. Projects include general pests, termite, active scents, as well as stewardship of Dow Agrosciences fumigant products. He was directly involved in the development and enhancement of the Centricon termite colony elimination system. As postdoc researcher 1992 to 93, field technical service and development specialist for urban pest management in the Midwest from 94 to 96, as a field biologist for early stage field research located in Jacksonville, Florida from 97 to 2002, and as field biologist for the East Coast located in Roanoke, Virginia in 2003. In 2004, Joe relocated to Indianapolis, Indiana, and was director of Dow Agrosciences Pest Management Global Laboratory for 4.5 years before taking his current position as field scientist and commercial biology leader for urban pest management located in Fayetteville, Arkansas, covering most of the U.S. In addition to his field scientist role, he also was the global biology team leader for all urban pest management projects for Dow Agrosciences from 2011 to 2015. Joe has been an invited speaker at numerous pest control associations and entomology meetings, including Kansas, Iowa, Missouri, Illinois, Ohio, Oklahoma, Colorado, Long Island, St. Louis, Memphis, and Michigan pest control associations. Hopefully soon we'll see him in the Texas. The Purdue, Kentucky, and Nebraska pest management conferences, New Orleans Termite Academy, pesticide training in Indiana, EPA Region 5 training in Indiana, EPA Region 7 training in Kansas City, the Entomological Society of America, the National Conference on Urban Entomology, the International Congress of Entomology, the University of Arkansas, and the National Park Service. He has authored and co-authored 16 refereed scientific journal articles, proceedings, book chapters, and several trade journal articles on urban insect pests. He also is an author, co-author on 21 patents, including the Recruit HD patent granted in 2015. In 2022, he received the ESA Southern Branch Recognition Award in Urban Entomology. He currently has adjunct professor status at the University of Arkansas and previously held adjunct status at Purdue University. He also was a 2005 president of the Ohio Valley Entomological Association. He has been a very busy guy. Joe, take the floor. Thank you.
All right. Okay. Well, thanks for that introduction. Yeah, I sent my mini bio, and I pretty much read the whole thing, but I appreciate that. A very nice introduction, and looking forward to presenting today to you on termite biology and baiting technology. And let me get my screen up here. I'm going to share. Let's see. And hopefully this will come up, and I'll get my. Is that coming up okay for everybody? Does it look good?
Yes, sir.
Okay.
It's in presenter mode. You look good.
Okay, good. Okay, so can everybody hear? Am I coming through pretty good on for sound? Okay, good. Okay, so I've got a lot of slides, but I have a lot of pictures too. So I'm gonna run through this. You'll see me run through it fairly quickly. I'm gonna get through a lot of material today, but I'll just start right in. Termites are actually, they've changed the order recently to Blatodiae, and that's actually the same order as cockroaches. And the old order, what it used to be, was Isoptera, and now that's an infraorder. Isoptera means equal size wings, and that's what you'll see with the swarmers, and that's where that comes from. So as I mentioned, they now are part of they're actually known as social cockroaches, the way they're showing them as far as their background and how they've developed over the years. They're soft-bodied, small social insects, live in colonies. That's very important for termite baiting and control, to understand colonies. They have a caste system, a division of labor within that. I'll go over that in a little more detail. Overlapping generations in the colonies, all different ages. Cooperative brood care is important when you're talking about social insects. They live sheltered, usually underground, sometimes in mounds, and there are mounds in Africa, Asia, even northern Australia. You'll see termite mounds, different species, but some of them will affect structures also, especially in northern Australia, the mound termites. They eat most anything, including containing cellulose, and they have organisms in their guts to digest cellulose. They require moisture and warm weather, and actually they are beneficial to the environment as wood recyclers or release energy back in the ecosystem. So they have some benefits breaking down wood in environments, but of course they're also major pests. You might get calls on what's an ant versus a termite, or I've got something swarming in my house, and they're actually really easy to tell apart. I think most everyone in this business knows that, but they have equal sized wings. Termites do, versus ants have two different sized wings if you're looking at swarmers. Termites have straight antenna, where ants have elbowed antenna. And of course, ants have a thin, narrow waist, while termites have a broader waist. So you can really tell the difference fairly quickly if you get called into a situation where there's a swarm, and determine what you're dealing with. So the approximate distribution of termites in the world are in this whole reddish, pinkish area here. And you can see it covers a big part of the world, all different species. The little hashes here are the Formosan termite and where they're found. Of course, they get into Texas, which you guys all know, I'm sure, dealing with them and along the Texas coast. For distribution around the world, there's over 2,900 termite species worldwide. Of these, less than 10% are actually considered pests that would get into structures and cause problems. In the US, there's about 45 to 50 species, and 5 to 7 of these are considered pests or serious pests. In the US, there's 4 general groups of termites. Subterranean termites, which are the main ones that are affecting structures, well, with the main getting the most damage. We got the reticulotermi species, including the eastern subterranean termite, or the native species, of course, very prominent in Texas, as well as other reticulotermi species. Also Formosan termites in the southeast US, including Texas and Hawaii. It's also a new invading Coptotermes, similar to the Formosan termite in South Florida, called the Coptotermes gestroi, or the Asian termite. Is just moved into South Florida. Don't really know what the range is going to be, but it's killing a lot of the trees and getting into structures down there, and actually out competing the Formosan termite. But the range is very small right now, mostly just up through Miami up to Fort Lauderdale, maybe a little bit further north than that, but not much further. Of course, there's drywood termites. I won't touch on those today. Dampwood termites, and there's also these higher subterranean termites that they don't molt as much, but they're not as much of a structural pest. Some of them are also known as Nasutotermes, have also come into Florida recently, but not a real big pest in the US. If you look at the pressure of the termites across the country, and it zeroing in on Texas, of course, you got very heavy and heavy for most of the state. You can see this kind of a line. Anything below that line is at least high to medium pressure to very heavy. You can see this yellow across the coastline, the southeast coast, including Texas, of course, is where the Formosans are, where they are considered the most very heavy areas of the country for termite pressure. So when you compare termite, the main 2 species, or the main types of termites found in Texas, you got the native subs, the reticulotermes species, versus the Formosan subterranean termite, or Coptotermes formosanus species. When you look at size of colonies, the native subs are, when they get mature, about 100,000 to 600,000. They can be even up to 1 million at some times, but this is on average. Whereas the Coptotermes, or the Formosans, you can get, on average 500,000 to 2 million plus. And there's been reports of 10 to 20 million in some termite colonies in in Formosan areas, but most of them fall in the 500,000 and 200,000 range, or 2 million range. When you're looking at soldier ratios, the native subs have 1 to 100, or about 1% are soldiers, whereas Formosans have 10% soldiers. So you're definitely going to see a lot more soldiers when you're dealing with Formosan subterranean termites. The soldiers of native subs are really non-aggressive, whereas soldiers of Formosans they can be aggressive. They actually will secrete a white fluid. It looks like glue behind their head, and they actually use this to fight off ants. But they will also, try to bite you, whereas the native subs really can't do that. Move this a little bit. And then the alate swarm, or the swarmers, are during the day from January to April for native subs. Further south, earlier usually, whereas the alate swarms for Formosans are April to July, peaking, usually in early May, early to mid-May, and they swarm in the evenings. So when you look at eastern subs or reticulotermes species, these are the main native species in Texas. They're the largest of the reticulotermes species, the Reticulotermes flavipes. There are other ones, Reticulotermes virginicus, Reticulotermes hageni, and a couple other species found in Texas. Not as big of a structural pest, although virginicus will get in structures, Reticulotermes virginicus. But they're really pretty much common throughout the U.S. and common throughout Texas. Formosan termites, when you're comparing Formosans to native subs, when you look at them, you can see right away that the head of a Formosan termite has more of a teardrop shape versus a rectangular shape for the native subs. And the soldier, or actually the workers, look fairly similar, but you find the soldiers, you can tell right away what you're dealing with. So what are the aspects of biology in termites that make them pests? Well, the main three things are their nesting habits, and their feeding ability, feeding on wood, and then their colony behavior. I'll go through some of this real quick. So their nesting habits, they're in the ground, they're in shelter, they're underground, and they have many different locations where they might feed. This is an example of a single colony that might get into a structure and be on a property. And I'll talk about how big their colonies can be here shortly, but and then you can have many colonies around a structure. So, just their nesting habits make them a pest, and that they, you don't know you have them until sometimes you have damage. Also, the ability to consume wood, and they actually have to have bacteria or symbionts in their gut, protozoa, that actually helps them digest the wood. But they'll eat the wood, they'll digest it, and actually after they molt, they have to regain these symbionts and bacteria in their gut to continue to survive. Another thing is their feeding habits is they have this ability to exchange regurgitated liquids, or going out feeding on wood, they can exchange it to each other. This is known as trophallaxis. This is very important to understanding how baits work, which I'll talk about here shortly, but they have that potential to transfer food within each other, but they also have the potential to transfer pesticide or active ingredients, which have to be slow acting and non-repellent so they can transfer them. And I'll go into a lot more details. We talk more about baiting. So detection, there's different ways of detecting. You got wood damage, extensive swarming in structures. Of course, that's a big sign right away. And then shelter tubes, seeing tubes or tunnels of termites. So, termite damage, if you're in the business, you've probably seen pretty bad damage over the years. When you're looking at subterranean, it's usually with the grain, and usually you see mud. And again, native subs are definitely like this. Formosans sometimes will cut across the grain because they're such voracious eaters, but you will see the mud with Formosans also. Whereas drywoods, you'll never see mud, obviously, and you'll see pockets of where they're feeding and where they're living in much smaller colonies. Again, termite damage, you'll see the mud. It's with the grain of the wood. Of course, extensive swarming, this is pretty intense here, but you will see this with Formosan termites. Not as big as swarms with native subterranean, eastern subterranean termites, but you'll see a lot of swarming and a lot of insects on the ground, swarmers in those particular situations also. But a lot of times, of course, the first sign might be a swarm, and you can go out and determine if it's an ant versus a termite, and then figure out where the damage is and do your treatments. And of course, baiting will, you'll be able to put baits out if needed in these type of situations. Shelter tubes, I mentioned, of course, these are also signs. Will be on the outside walls, inside, in many cases, and this is what you look for. Of course, that's a telltale sign. Break the tube, and most likely, if it's active, you'll see soldiers come out, especially for formosans right away, and you'll see some termite activity in these. What are their environmental requirements? What do termites want? What do they need? They need food, of course, wood to survive the colonies. They need moisture, and they need shelter, or anything, could be a tree for formosan termites, to structures, to any kind of shelter where they usually build colonies underground and into wood. So what's conducive to termites? Of course, wood to ground contact. Termites, they need food, water, shelter, but if you have an opportunity for them to get in real easy with wood to ground contact, they'll easily build tubes up into a structure. Very conducive. Of course, wood debris against the wood piles against buildings are bad sign for, especially in a termite area, to see. I always tell your customers, move that away from the house, of course, but it just gives them, a conducive condition for termites to get into a structure. Any kind of wood debris around the house can cause that. Even vegetation, which eventually drops wood debris next to the house, but also keeps a lot of moisture in and gives them a lot of shelter for termites to survive around structures. Of course, mulch is another biggie. People use excessive amounts of mulch around their house. Some of the untreated mulch, they might get free from the city or something. They'll say, "We'll get free mulch, come get it," and put it around their house, and they're just putting wood around the house and inviting termites to come in. And then, of course, moisture issues. Got to get the moisture away from the homes, and moisture can cause termites to come into the homes, or at least stay around the homes, and then get into the structures from there. Of course, poor ventilation also can contribute to termites. Again, they need moisture, and poor ventilation in crawl spaces, basements, other areas can be conducive to have termites want to come in and get into structures and stay in structures. Moving on, and I'm kind of going fast, but I got a lot of material here. I want to talk about getting into the termite life cycle of termites. And so the termite life cycle, you might have seen this before, but termite colonies start with swarmers. They swarm from colonies. Swarmers, also known as alates, the winged termites, will fly from a colony. A lot of times they'll fly all at the same time, the different colonies, and then they'll pair up. Alates will find a mate, male and female, pair up, and they'll shed their wings, and they'll look for a place to start a new colony. So what do some of these swarmers look like from some of these different species? You got formosan termites, the largest of the swarmers of the native or the more structure infesting species, but they'll be tanner in color and larger. And again, these swarm at night, versus some of the other species here, particularly Termes flavipes, the native subs, dark black, dark brown to black in color, smaller than the formosan. Again, these will swarm during the day, usually after a warm morning, after some kind of moisture in the air. Virginicus, very similar, but smaller. That's the southeastern subtermite. Again, all these are found in Texas. Hagani is a little different. It's smaller, Termes hagani. Not a real big structural pest. You'll see them more in native, in wooded areas more. These will swarm in the spring and in the fall, and they're all tan in color. So moving on, the new king and queen, as I mentioned, they'll mate and they'll produce eggs and start a colony. So what do these kings and queens look like? Well, the drywoods here on the left are usually more darker in color, whereas the And the native subs here on the right, they get the main primary queen who gets physogastric and just basically filled with eggs. And the king, when they first start, and that king's always going to be smaller over time than the female. And the king's really just there to mate with the female through its entire life. The female's producing eggs every day, laying eggs constantly, building up colonies. It's a young colony of actually Coptotermes, taken by Thomas Chavanc, who's at University of Florida. You can see a very young colony that he came upon. You can see some eggs and some young termites growing in the colony, soldiers and workers starting. This was from a picture that it was sent to me from one of our territory sales managers in, actually, this was in Missouri. This was, you can see like a hose here. They pulled a piece of wood, and in the wood was a very large termite queen. You can see the king here. And if you take a look at the close-up, this is kind of what they might look like. This is very rare to find these in the field, especially for native subs. But you can see this queen, very physogastric. In other words, just filled with eggs. And the king here is right next to it. And then you can see some workers tending to the queen. Here's a soldier up in this area here. But really, really cool to see that in nature or in the field or in a structure. If you see it, you don't get to see that very often. Sometimes you never see it in a lifetime, but they're really hidden away in structures. So these eggs hatch, and the eggs turn in. The first molted casts are called larvae. The really young termites are called larvae, and then they molt. And molting is really important for baiting. You got to know that termites are molting over the years or over the months, and they average, a termite averages a molt every 45 days. However, the younger larvae molt much sooner than that, and as they get older, they don't molt as much as that. But on average, it's 45 days. You'll get a turnover of all the termites in the colony will actually go through a molt. And then these larvae, as they go through several molts, and there's usually, the native termites, it's usually about 7 to 9 molts to become adult workers, soldiers, or what they call nymphs, which are going to become your alates. Again, as these colonies mature, they will leave the colony and start new colonies again, and the cycle continues. The eggs you can see in a colony, there's usually in bunches, and they usually have soldiers hanging around protecting the eggs. You can see the really young termites here, the larvae in this area. And there's a main nest usually in every colony where the main king and queen are, and this is the brood of that colony. The larvae, again, the real small termites. If you find really small termites and you find eggs, you pretty much found the main source of that colony. And the king and queen are not far behind usually in these areas. And then, of course, the workers are, the termites you'll see out there in the structures, feeding on structures, and you'll see them going in the tunnels. They're the ones doing the main work of the colony. They go out, get the food, feed the soldiers, take care of the soldiers, take care of the king and queen, take care of the young. So they do all the work. And then the soldiers themselves are there for protection. And they might go out with the workers foraging for food for protection. And then nymphs, as I mentioned, these nymphs will molt into alates and take flight and start new colonies. One thing I haven't mentioned here are supplementary reproductives. You don't always see these in colonies, but if the main king or queen will die, some of these adults will develop into supplementary reproductives. They look larger than workers and nymphs that have these wing buds on them before they become and they get in the colony. And when this happens, and sometimes really mature colonies will get these, the colonies can really grow because they'll also be laying. They'll have multiple supplementary reproductions and they'll all be laying eggs throughout the colony. And these kind of look like this here on the left, secondary or supplementary, they're called. And on the right here, they're larger than the workers, and then within colonies, they'll just grow the colonies very quickly. So I mentioned this division of labor. It's a caste system. You got your reproductives, both the male and female, in the colony and the nest. They disperse. The reproductive alates will disperse, or swarmers, establish new colonies and develop, and they will also develop into replacement reproductives or supplementary or secondary reproductives. So There's different casts here of reproductive in colonies. Again, starting mainly with the main king and queen. And these main king and queens can live, you know, at least in the literature, they say live up to 20 years or more even. They can live a long time. And then if they die, then these replacements will take over. Of course, the workers I mentioned, forage, feed, and groom, take care of the young, and sterile or immature, they do not, you know, they can't have young or anything, and they don't mate. It's the main king and queen that do all the, you know, laying all the eggs and growing the colonies in the replacement reproductives. And then the soldiers are mainly for colony defense and initiate foraging, going out with workers to do that. So I've talked a little bit about colony. So what is a colony? You know, and termites tend to group in colonies. Termite colony is from an individual king and queen. They'll start their colony. It's grouping individuals that eventually will share a common system of tunnels and nests. They'll freely transfer food among themselves within the colony. I mentioned trophallaxis, how they feed each other. They cooperate in rearing of the offspring of a small number of reproducing individuals. So they'll just, you know, within that colony, they take care of each other. They cooperate and maintain the colony infrastructure, to build the colony out. And I'll talk about foraging and that as we go along here. And they maintain their integrity with respect to other colonies. A single colony, even if the same species, they don't necessarily mix. They don't mix together. So you'll have one colony. You might have several colonies around one structure of all different ages. And then, you know, when you can take out colonies with baits, which will go into more detail on that. So what are the colony dynamics? I touched on some of the sizes already a little bit on these different species, you know, 100,000 to 200 million plus, depending on species, but also how big can they forage, or how big can these colony areas be? And they can get really, really big. You know, you've probably heard from some people say, oh, they can be as large as a football field. And that's true. And they can be even larger. 450 feet is in the literature. I've actually seen, and I'm going to show an example of a colony that was as big as a football field of a Formosan colony. Factors that influence colony size, of course, the age. The older the colony gets, usually with the more mature, multiple reproductives, over time that colony becomes bigger and bigger and spreads out to a bigger area. Also factors that include resources. If they have enough food that's palatable, or a lot of wood in the area that they can feed on, that they find palatable, or you know, what they want to feed on, and then water in an area. These are all critical. Moisture, as I mentioned. Of course, factors, species, as I mentioned, Formosans could be larger colonies than native subterranean. Then, of course, geography, where they're found in the country. You know, along the Gulf Coast is big for Formosan termites. So how do you determine colonies? And we did this years ago. And just an example, how do you determine a colony area? When we did mark recapture years ago, and this might be a plot. These green circles could be a bait station with wood, or they could be a bucket trap. When we first started doing these with bucket traps, bucket in the ground with some wood, the termites can find, you put these out in a plot area. And then you find these little arrow hairs are where you come back after a month or two and you check all these bucket traps, or all these could even be stakes. Which ones have termites? So you find which ones have termites, and then you pick one, and you might dye it a certain color, like blue. For instance, you dye the termites. So you take the termites back from one of those traps, you clean them up, you put them on dyed filter paper, and they turn blue. As you can see, you can turn them blue, you can turn them red. I've even seen green. There's different colors you can use with these dyes. Doesn't hurt the termites. They just get dyed blue, red, green, purple even. You re-release them in that original spot, and then whatever ones show up, you know that's the same colony. Like these ones out here might be different colony. You can actually do this about 3 times. So now you know which ones are blue. It's all the same colony. You might pull termites from all was there 5 of these, bring those back, then re-release all the termites back, and then see if they've spread out even more, and you can do this like up to 3 times. This can show you the colony area, and then you can determine how big these colonies can be. And then you could do like a mark recapture, Lincoln index procedure that'll kind of give you a ballpark estimate of the population. And that's where they come up with these populations of 2 million termites or 100,000 termites, looking at the ratio of the blue termites they find in these bucket traps versus the non-dye termites. And then what's really been good to determine colonies has been DNA studies. So that gives you more of an area and which ones are within an area that they mark recapture. DNA can't really tell you how big an area, but it can tell you what colonies are, if they're different colonies. So, they've opened up a whole new understanding. We use this all the time now. We don't even do the mark recapture much anymore unless we're trying to figure out how far spread out they are. But you can also use the DNA to find them in a certain location in another location to tell you that area. But you can usually pick about 10 termites, and these are put into a procedure with a gel, and you separate out the DNA, and you can see at one colony from another how the DNA is separated out, the Gen X are separated out. So colony 1 is on the left here, and colony 2, you see the Gen X at the lower range of this on the right. So this is really an easy way to determine colonies now. And for an example here, there's some trees, and I'm going to go into more detail at this site but there's 1, 2, 3, and trees 1, 2, 3, and 6. Tree 1 and 2 at a location, or tree 1 had the same colony, but 2, 3, and 6 had this. We're all the same colony, and you can look at the genes. And here you can see this is different than over here. And this is like really, really accurate as far as determining what colony is where. And so now it opened up an opportunity to look at this. Dr. Ed Vargo is at NC State, who's now at Texas A&M. This is an old slide from him. He does this in Texas too, but he's a real DNA expert. I've gone to him. In fact, for all the Recruit HD work we did, he did all the genetics work for us to determine colonies around structures and if we were eliminating colonies. But this particular site, he matched up, just looking at the DNA, these different colors and centricon stations that were hit were all different colonies. There were up to 10 different colonies. I've actually seen up to 30 colonies around a structure in Mississippi, and also another structure in Tennessee. So we know that multiple colonies can be around a structure. When we did our work for Recruit HD, we did 111 baited structure trials. 66 of these trials sites had at least more than one colony or more. 41% had one colony, 29% had 2 colonies, but you see, and we had a couple sites that had up to 13 colonies. Like I said, we have had now sites up to 30 colonies. But this gives you an idea. There's a lot of colonies around structure, not just one affecting structures. There might be a couple at the same time. And if you look at Texas, these are the efficacy numbers of the numbers of colonies per site by state when we did Recruit HD research. Texas averaged about 2.1 colonies for native subterranean or reticular termes flavipes, and about 2 colonies of formosans around all the structures. I think we had, not a ton of a colony and not a ton of structures here. Maybe, I forget, it was like 6 to 8 or something in Texas that we looked at, but they average around two colonies per structure, depending on species. Now let's look at the foraging pattern. This is very important to understand for baiting also, to understand how termites forage and why they hit termite bait stations around structures based on their foraging patterns. So in general, subterranean termites tend to distribute the intensity of their attack over multiple targets. So they're always foraging, and their quality and quantity of their food affect their feeding preferences. Termites move continuously among feeding sites, and they tend to move in groups. like raising animals, as I've heard people say, but they tend to move in groups. When a single termite finds a location, then they mark it and they get other termites to come back to it, and now it's a feeding location. They move in groups to these locations. This is some work that Dr. Nanyao Su did at Florida in a two-dimensional arena. He put it in there's sand or soil here, and you can kind of see he's got a two-dimensional arena where he's got like plexiglass in between, and he releases termites in the center, and he looks at how they forage out from this center. This is an example of a close-up. He releases termites here, and how do they forage? And what he sees, and this is for most of termites, it's like they start branching out, and as they go out, they branch more. It's like branching on a tree, and they actually looked at this in a lot of detail. The termites will branch out as they leave and from a main colony, like branches on a tree. They're called fractals, but they've looked at this. And if you look at a tree, how many branches there are, that's kind of what you're seeing underground with termite tunneling. And this is Flabopes. They actually have, if you compare it to Formosans, tend to have thicker colonies, not as much branching to start with, but this will pick up, whereas Reticulotermes flabopes tend to do thinner colonies, or I'm sorry, thinner tunnels. But they will, when they do find food, they'll make them thicker in different areas, but they'll forage on thinner tunnels. But you can see how intense the branching can be. This is usually just, you republished this back in 2001, but gives you an idea. They find wood here in this, and then they start working on that. So looking at this diagrammatically, they first branch out slowly, maybe send out 4 branches, and then they branch out even more over time, and then even more over time. So, it kind of looks like a tree underground. And then, you'll see thicker, different ones. And then when they do find, let's say they find food up here, and they will all of a sudden, when they do find food, like you put a piece of wood here, this is not food, this is not food, just as an example. They'll find food, and then they will put down a pheromone trail to that food. So termites have pheromones. They'll mark that food, and then they'll concentrate in that area. They'll send out signals, colony-specific signals and scents to that area, mark it, and then the termites will start foraging there, and they'll actually build that tunnel to that food or to that area, and mark it. That's what they're doing in the real world too. They find a piece of wood in the ground or find a structure, they'll mark it, and then they'll get the main colony will zero in on that area. But they continuously look for food in all different areas, so they're constantly moving. So when you look at this kind of a pattern here, in this over time and how much they tunnel, termite stations are on average about maybe 8 to 10, 12 feet apart around a structure. They will find them. Termites are there, they find these stations. And that's why, what about the gaps in between stations? People ask, well, they're going to find them because this is how they forage. So again, just diagrammatically, they leave the nest, they'll find a wood source, they mark the wood source with a pheromone, go back to the main nest, let them know after they mark it, and then you'll get multiple termites going to it. And this could be a bait station, can be a structure, it can be whatever they find, and then they'll you'll see them. As I mentioned, I mentioned that I wanted to talk about this site, that it's a Corteva New Orleans mosquito and termite and rodent control R&D site I did in New Orleans. And this showed how huge a colony there was. This is as big as a football field. These trees, 1, 2, 3, and 6, all had termites in them. And we took the DNA from these trees and determined which one were connected. We thought, well, maybe 1 and 2 would be connected, maybe 3 and 6, or maybe these will be separate. But what we found is, again, I showed this earlier, tree 1 was all by itself, 1 colony, but 2, 3, and 6 were another colony. So this is an area, and from here to here was 340 feet. So we're talking about a length of a football field here. And this is just what we know. They could actually even be further than this. But DNA showed that these were all the same colony. This was a separate colony over here. And if you look at it, here's the tree 3, and you look from 3 to 2, that's what it looks like. And it's behind like a shed here on the property. That gives you an idea how far these termite colonies can go. And this is the other from tree 6 to 2. So that's that whole area. There's even like a restroom area here. Again, this is Fort Pike. It's a national fort. So a summary of this, we found this colony be bigger than a football field in length, and actually we baited one bait out there and we eliminated the colony, 340 in length with Recruit HD. So I've been talking about biology mainly. I'm going to move into baiting here more, and why termite biology is so critical in using baiting technology. The exchange of food between members. I talked about trophallaxis. That's important to know because, termites forage, continuously feed at random sites, and then they feed each other. So if they're finding bait, they're going to feed each other that bait. Again, trophallaxis. And what we've seen, and I can show some examples of some papers that have been out there, really only need one station of a bait with a chitin synthesis inhibitor, and I'll go in more detail how they work, to be hit to get control of an entire colony. Baiting takes advantage of the natural termite behavior, this continuously foraging, looking for food, and that foraging pattern. Active ingredients. The active ingredients that work, I'm not going to go on the ones that don't work, but chitin synthesis inhibitors are the ones that work. And they're the ones that affect termites at the molt. And as I mentioned, termites molt 7 to 9 times. They molt on average every 45 days. So they're constantly molting, they're feeding each other. And it's important that it's not dependent on the dose amount. Chitin inhibitors only kill termites when they molt. So they feed on it and they don't die right away based on dose. They can eat as much as they want and they don't die until they molt. So they can feed each other, for 45 days or more, this bait, and then eventually, until they molt and then they'll die. So these baiting works at just chitin synthesis inhibitors. I'm going to say CSIs on some future slides, but they affect the termite molting process. They kill them at the molt. They also disrupt reproduction, and eventually the colony is eliminated. You talk about how we're doing, and I won't go into a ton of detail, but you put out termite bait stations. Labels say not to exceed 20 feet. We recommend about 10 to 12 feet, 8 to 10 to 12 feet, depending on the situation for Centricon. We recommend not installing them within 18 inches of the foundation because it might have already previously been treated with a soil termiticide. If a soil termiticide is there, it's probably not. They're probably not going to get hit if it's right in the soil termiticide. It's also, there's been some work done recently not to bait and use liquid termiticides at the same time, because what happens is when you're using liquids, you get these dead zones where there'll be some termites dead. And if the termite station is close, and then when these termites die in an area, termites tend to wall these areas off and stay away. So you may not get as good a hit. So if you're using baits, our recommendation is you just use baits without liquids, although you can just keep them farther apart from each other. So if you're using a liquid, we don't recommend it, but if in the situation that happens, just don't put it real close to where the liquid's being used. There's other things here, but basically you put them out every 10 to 12 feet. They're going to be found based on that foraging activity. Chitin synthesis inhibitor baits. As I mentioned, termites will feed each other, trophallaxis, they'll feed on a bait, feed each other, stops the molting process. As workers die off, colony declines, collapses. Actually, it's interesting, there's been a lot of work done that these termite workers that feed on the bait, they actually go back to the main nest and molt, and they die in that nest. And then eventually, as these nest areas die, they'll move the queen out of these areas. It's kind of interesting, the biology of that, until eventually the whole colony collapses and they die at the molt. They get stuck in their skin or their exoskeleton and they die. Characteristics of baits, they help ensure the toxic, you have to have delayed action. Again, they don't die till they molt, so that's delayed. cannot be too fast because they won't spread it around the colony. They don't die. Termites die, but not at the bait location. Again, as I mentioned, they go back to the main nest. That's why you don't see a lot of dead termites usually until right before the colony collapses. You might see them in above ground sometime. And that's because that's probably the last area that they're molting and dying. Formulation must be palatable. They need to be a bait matrix that they'll want to consume versus, say, wood in a structure or wood next to a structure. You want to make sure that they can compete. The bait matrix is very important. And again, some bait systems, Crude HD for one, are baited all the time now, always active in the ground. The mode of action, as I mentioned, CSIs, they kill them at the molt, and you can see they get stuck in their exoskeleton and they die. And these are some pictures we have. They also have ovicidal properties. So the queen gets fed. Queen's not molting, but it gets fed the bait from workers coming back. And the eggs that she develops will be exposed to the chitin inhibitor. And those eggs will not hatch. The eggs start to develop, and then the young within the eggs do not hatch because they don't develop an exoskeleton, so they become dead eggs. And then, so that's another property. You get molting, dying at the molt, and then eggs not hatching. And then they also have some other effects. Sometimes it's been mentioned that chitin inhibitors can affect the gut and tracheal system of termites. Real quick, baits were developed in '94 by Dow Agriscience. That's when Centricon was launched. FMC came out with sulfuramide, a slow-acting stomach poison, in '96 in a cardboard that was discontinued. You don't see that anymore. That was too fast acting, really. It was stomach poison, but they knew what was happening, or they didn't know, but they would get sick in an area, die in an area, and then they would stay away from it. Other companies came out with other baits with diflubenzuron, which is a chitin inhibitor, chitin sensitive inhibitor, and hydromethonon was actually out for a short time. BASF introduced an advance with diflubenzuron in 2003. Dow Agrisciences, we moved to always active with Recruit HD, eventually with novafluoron in 2011, and then BASF has their Trelona introduced in 2014. And there are several different, in addition to what's in the US, on the left here, these products are in the US and additional countries. And then outside of the US, there are also a lot of different bait products. Pretty much all these, I believe every one of these is a chitin synthesis inhibitor bait. So we've done a lot of research over the years, continuously trying to improve Centricon system. I use Centricon as my example because I've been working on it over 30 years. But we've come up with many different things over the years, new AI, novafluoron, bait mix improvements, station improvements. We've got all these different things. I won't go into a lot of detail here. We actually evolved, you can believe it or not, what some of these stations look like when we first started working on them to what's here on the right. Of course, we went through different wood monitors and electronic sensing protection over the years, but with wood, but we've gone now to always active with a bait in the station from day one. And here it looks at the different baits over the years. We started with a sawdust type bait, went to paper towel. These were with hexafluoron, went to novafluoron with paper towel, novafluoron Recruit 4 with the briquettes bait, and then now we have Recruit HD, which is our main bait for in-ground. Again, I mentioned how important termite bait matrix development has been over the years. It's got to be something that termites really want to eat, and it has to be durable. It has to last in the ground a long time. And that's what we did with Recruit HD. It's very durable, lasts for years, and termites find it, and they find it very palatable. They want to feed on it. Here you see just some example here on the right, taking out a station with termites and how they're just devouring the Recruit HD because they find it very palatable, and they want to feed on it even more than wood. And we did some work where we looked at blank bait versus wood in the field, and in every case at 90, 180, we looked at these quarterly, and we always had more feeding on Recruit HD versus wood at the same locations. Some advantages of baits. They're less toxic than most liquid termiticides, in the environment. There's less pesticide apply. We're talking about grams versus gallons here. Baits target a pest. They're there to target termites, nothing else. So we're not killing a lot of, non-targets. Have little or no impact on groundwater. I can say Recruit HD does not leach. We've done all the studies on that. It doesn't move out of the bait stations. Little or no impact on wildlife and the environment. You don't have to worry about getting into lakes or streams or wells. Usually no destruction or disruption to structures. And, I say possible elimination. We know they eliminate colonies depending on the product. But chitin inhibitors do a great job eliminating termite colonies. Some more advantages, but can use it as a preventative or a curative treatment around structures, good in chronic retreatment situations, good for spot treatments, for example, above ground baits, and I'm going to talk about that here briefly. Can be used with other treatments. Don't always recommend them putting close to liquid treatments though. A negligible risk in sensitive situations and valuable in areas of difficult construction. So we first started developing Centricon. We said, put them out in your toughest spots and where you really couldn't do a lot of liquid or your chronic retreatment situations. And it's a whole different mindset, targeting a colony or colonies around a structure versus, trying to keep them out of a structure with a liquid. Some disadvantages, some products have limited independent research to demonstrate efficacy. I can say Centricon, we've got, multiple publications and information on it. Too much moisture, too little moisture can cause problems. Initial costs can be high. I think that's come down over the years with this always active and only having to check annually. Repeated site visits, not as big an issue with our annual labels, and potential, there could be a potential for time to first hit, but with always active now and knowing how, because the baits out there are not just wood from day one and other baits that are out there from day one, it's not as big as a disadvantage as it might have used to be. How much bait of a chitin inhibitor bait do termites actually need to consume? It's very little. There's been some papers that have come out. In fact, 2021, year and a half ago or so, showed that only 5% of workers feeding on a chitin inhibitor bait, would they use Recruit HD for 5 days, was sufficient to eliminate the colony. I believe it wasn't a really big colony, but then they extrapolate out, that like up to 1 million termites, that it's very small numbers of termites feed, and they feed each other, and then eventually it gets into the colony collapses. True threshold is likely between 2.5% to 5%, what it mentioned in the paper. The minimum threshold of Recruit HD consumption needed for colony elimination is estimated at about 15.3 grams per million termites. So, that's not much. When you think about a Recruit HD bait as 150 grams, it's only 10% of a bait can kill a colony, based on this work done by Tomas Shavank in 2017. And then he also had a paper that colonies which fed it for a single day consumed as little as 1.1 grams of bait, or 5.5 were eliminated with 90 days. But I will say this was a lab colony of around 60,000 termites, but he replicated that. And then he extrapolates up on larger colonies. And just to wrap up here, we're up to about 50 minutes. I want to wrap up here in the next couple minutes, but I want to at least touch on above ground baiting at the end here. Recruit AG Flex Pack, flexible station components. If you're familiar with the product, we have a beige hard cover, and we also have a combination, or you can use a flexible cover, and there's a flexible pack that you will actually put where the termites are. The black flexible cover with a flexible pack maintains flexibility of the station so you can use it on uneven surface in and around corners, whereas the hard cover gives you added security if you need it, maybe in a high traffic area. It also allows for stacking, although I don't see people stacking these as much as they used to anymore. But it does allow, you could stack station on top of another station on a wall, say, and get more bait into an area. Above ground baiting, again, the flex bait used on any active infestation or swarm site. And it works on a swarm site, but you have to be real careful of the timing on that. You got to get it really quickly after a swarm. If there's a swarm tube that's been, termites have left that area, sometimes you don't get as good a hit rate on swarm sites. But active infestations, tunnels, activity areas, if you put them right over these areas, they find the bait to be, highly palatable, and they want to feed on it, and they get in these and they'll consume it quickly. 100 grams and 0.5% no fluoride. It's low impact. You're not drilling into structures too much. Direct baiting for immediate termite consumption. Successful placement speeds elimination. I recommend using these wherever you can. Unique targeted approach can be used with the in-ground system, or can be actually as a standalone. Most people just use them with in-ground, but in a situation, the label does allow you to use them as a standalone, but you have to check them quarterly. You can't be out there just once a year. And really, to use them correctly, you should be out there every 3 to 4 weeks checking them. The matrix prep for baiting in approximately 1 minute. When I say you should be out there every 3 to 4 weeks checking them to stay on top of it, but by label, if you use in-ground, you don't really have to be out there for a year, but I recommend getting out there much more than that to check them. So recruit for moistening, you'd probably cut it. There's a line here, example, you'd cut it and then you'd add about 3 to 4 ounces of water. Knead it. This is before we actually became a product. I got some pictures here as experimental sampling. Knead the bait, becomes like paste-like, and then you install it. And again, the flex station can be installed on different uneven surfaces if you need it. And it actually will allow for narrow areas where sometimes you might have to dig out an area. This is a structure in Arizona that they put some up. They actually dug out an area with the hard case, but they didn't have to do that with the soft, flexible case. I'm going to mention just a couple field trials here, and I want to talk about what we did in Texas a few years ago. We just picked sites. We did above ground only. We installed 1 or more termite. If we had 1 or more active tube or damaged wood, we installed AG stations only. We put up multiple stations. We used both soft and hard cover. We took termite samples for DNA to identify colonies. In case termites came back after we thought we eliminated, make sure it was not the same colony. So we do this all the time. We take DNA just to have background. Replaced if needed, and then we collected data monthly on hit, termite numbers, consumption. Here's an example of a site we did in Lamarck, Texas, near Houston's Formosa. Above a porch, we put one of these stations up before I put the black cover up. Took a picture here as an example, putting it on an uneven surface. This is above a porch with some, decorative woodwork here. And I'll just show this real quick. These were 4 sites we did in Texas. Lamarck and Beaumont were Formosan sites. These are AG sites only. Galveston, and believe it or not, Galveston, we had Reticulotermes flavipes, and we had Texas A&M site building that had flavipes. But the blue bars here are months to elimination per site. So Lamarck took us 3 months. Beaumont took us 2 months to eliminate termites, and the mean average was just under 3 months. The amount of bait they consumed is based on what's here with this red line. For example, Lamarck ate 600 grams of bait, or approximately six of those above ground bait stations are 100 grams each. You can see less here for some of these sites. This one a little over 2 at the Texas A&M site, just as an example. But we average a little less than 3 months for elimination. Again, these are above grounds only. And we averaged a little between 400 and 500 grams of bait consumed. And again, we just put these up everywhere we could. This was Lamarck site. There were some piers out in front of the site. I took this picture after 1 month of feeding. These were full. We took a picture. They ate the baits completely out. And this site was eliminated in 3 months. This is that site, same site above the porch. This is what it looked like after 1 month that we baited. This was a Galveston, Texas site, 1 month, Reticulotermes flavipes. Put these up, and the baits were completely consumed. This surprised me because this is flavipeas. Took these down, put 2 more up. They were completely consumed, put 2 more up. By the 3rd month, they ate a little bit more and the colony's eliminated. So very interesting. That's all I got. I don't know if I have time for any questions, Jesse or Andy, but wrap it up right there.